Hello friends. In the previous lecture, I tried to give the brief about the fatty acid synthesis. Today I am going to give some thing about the triglyceride synthesis. We try to learn about the triglyceride synthesis and something related to disorder. We try to discuss the how the triglyceride synthesis get affected in a well-fed condition or in fasting condition, and as well as the most prevalent disorder, which is diabetes mellitus, in which how it get affected, that we're going to discuss. Before starting with the triglyceride synthesis, I'll let you know some basic about the triglyceride, and then we go about its synthesis pathway. We all know that we having the different type of storages for the energy. Like for the glucose, we having the glycogen, or we was like I say the glycogen is a storage form for the carbohydrate. Skeletal muscle is a storage form for the protein. Same way triglyceride is a storage form of the fat, and that fat is mainly stored into the adipose tissue. And this particular adipose tissue is stored with the mainly with the subcutaneous tissue, and in many part of the our body. We will discuss later that part. Here you can see, when you think about the triglyceride, triglyceride means tri means three, glyceride means it's related to glycerol. So this is a glycerol molecule. It's having the 3 carbon. When this 3 carbon compound with this OH group, it means it's considered as one type of alcohol. When it's bound with the fatty acid, which is a lipid molecule, so we can simply say that triglyceride is alcohol plus lipid molecule will bound together and form the triglyceride. This fatty acid can be any one. It may be a saturated fatty acid, it may be unsaturated fatty acid, it may be a PUFA, it may be a MUFA. So what type of fatty acid we are taking in a diet, according to that, fatty acid will be bound with the glycerol, and that type of triglyceride is there with the our adipose tissue. Now again I am come to the adipose tissue, and before we discussing about the adipose tissue, I just give you some practical point. Many of us might be attending the gym and trying to lose the weight, or sometimes we're trying to gain the weight, and we are searching on the internet. You will find this type of figure which shows the body fat percentage. What we can see here, the different persons are there. The different body structure is there. Some looks very muscular, some ones look very fatty, someone is a middle in between. And when we compare it, we can see that this particular person having the very less fat percentage, that is only 3 to 4%, and that is why the each and every skeletal muscle is visible. When we come to this extreme part, the person who having the 40% of fat, it doesn't mean that he doesn't having the muscle, but we can say whatever the weight is there, from that 40% weight is of fat. Why we are discussing here? It means we have a fat, only difference is that percentage. And when the fat is there, mainly in a male, there is a truncal obesity is there. In female, the obesity is there with the buttock and mainly with the thighs. So there is a difference between the percentage of fat. Now it means whatever the triglyceride is there, which is stored in adipose tissue, and that adipose tissue is present everywhere in a body. Okay, everywhere in a body. Now to the next point. So always it will raise the question in our mind that does the triglyceride having a direct relation with the obesity? Is it a different one or is it the same one? So it's a very logical thing that we have to understand. When you see that the increased triglyceride level in the blood, so when you think the increased triglyceride level in the blood, it means there is an increased circulatory triglyceride, or you may say that it indicating the circulating chylomicron, which is one type of lipoprotein and which composed of 98% with the triglyceride. So when the increased triglyceride is there in a blood, it means there is a circulatory TG is more. And it doesn't mean that we having the more triglyceride in adipose tissue, and because of the more adipose tissue, that person having the obesity. So to understand, this increased TG or increased chylomicron may be remain high for the few hours after the food ingestion. So when we taking the food, and if our food having the more amount of the fat, then for the few moment when we eat it, then after triglyceride and chylomicron will remain high in circulation. As well as if there is some metabolic disorder is there, when the triglyceride metabolism is get affected, in that case also the triglyceride may remain high in the blood. So this is about the circulatory triglyceride. It doesn't having any relation with the obesity. Same way, when thinking about obesity, it means more body fat percentage or adipose tissue, which is in a peripheral tissue, which is a store form, which is not there with the circulation. Okay, so it's a increased storage of fat. Circulatory may be a normal one. So if the person is obese, does not mean that there must be a high triglyceride level. So you may come to know that when person is very thin, he might be having the high triglyceride level, or you may come to know that patient is obese and having the absolutely normal triglyceride level. So that way to understand that's this topic. Now we come to the main part of the triglyceride synthesis. There are the three precursors for the triglyceride synthesis. One is glycerol 3-phosphate, dihydroxyacetone phosphate, and monoacylglycerol. This glycerol 3-phosphate we all remember when we learn about the glycolysis, and when this glucose is broken down and the glucose is converted to glucose 6-phosphate, then fructose 6-phosphate, and fructose 1,6-phosphate, and then after it is broken down to the dihydroxyacetone phosphate. That dihydroxyacetone phosphate may convert it to glycerol 3-phosphate. So that may be a one source through which triglyceride gets synthesized. Sometimes dihydroxyacetone may be one of the precursor with the same phenomena, and it will help for the synthesis of triglyceride in liver and adipose tissue. Third one is a monoacylglycerol, because most of our food has a triglyceride as a fat. So when we eat it, when it goes in digestion, almost 65% of the molecule which come after the digestion of the fat, it is monoacylglycerol, and that monoacylglycerol comes into circulation, and once it comes into circulation, or better way you say go to the lymphatic tissue, then it goes for the formation of the triglyceride. Now we go with the reaction. So the basic idea is that for the synthesis of triglyceride, either glycerol is there, or glucose is there, or dihydroxyacetone phosphate is there. It has to be converted first to the glycerol 3-phosphate. So So that is why there is a two enzymes are there. One is a glycerol 3-phosphate dehydrogenase, which convert the dihydroxy acetone phosphate to glycerol 3-phosphate. And there is a glycerol kinase, which convert the glycerol into the glycerol 3-phosphate. Sometimes when we eat the fat, when the fat is goes on the breakdown, from that also we are getting the free glycerol molecule, and that glycerol molecule also converted to glycerol 3-phosphate and ultimately utilized for the triglyceride synthesis. This glycerol kinase enzyme is absolutely absent in adipose tissue, and and that is why in adipose tissue major triglyceride is synthesized by the dihydroxyacetone phosphate, and this enzyme is active in a liver. So once because of the glycerol or dihydroxyacetone phosphate, glycerol 3-phosphate gets synthesized, then this type of sequence of reaction will happen and triglyceride gets synthesized. We are not trying to concentrate over each and every step, but try to understand that glycerol has a 3 carbon. On the third carbon there is a phosphate is there, and when it goes in a sequence of reaction, you can see 1, 2 and 3. By sequence of this reaction, 3 fatty acids are added. So one by one is added, and glycerol and phosphate is removed, and that glycerol is converted to triglycerides. So in the first step, by the glycerol 3-phosphate acyl transferase enzyme, fatty acyl CoA will bind with the glycerol 3-phosphate. CoA is removed, and the glycerol get the one fatty acid which is bound with the first carbon. So in this molecule, lysophosphatidic acid, it's the same like glycerol, only thing is on its first carbon fatty acyl CoA is there, and on a third carbon there will be a phosphate is there, and that molecule we call it is a lysophosphatidic acid, and this enzyme is called a acyl transferase, because it's just only fatty acid get transferred from fatty acyl CoA to glycerol 3-phosphate, and that is why enzyme name is a glycerol 3-phosphate acyl transferase. In second reaction, same is going to happen, which I told you. This lysophosphatidic acid also we can tell 1 acyl glycerol 3-phosphate, because first carbon fatty acid is there, or third carbon phosphate is there, ultimately is a glycerol. So enzyme is a first acyl glycerol 3-phosphate acyl transferase. Again fatty acid is added, and now on that molecule we call it is phosphatidic acid. Okay, so in this phosphatidic acid means now another fatty acid is added, that fatty acid is added on a second carbon. So now in a phosphatidic acid there is a 2 fatty acyl CoA is there. First already was there with the glycerol. Now on a second carbon another fatty acyl CoA got added. On a third carbon already phosphate is there. So this is called a phosphatidic acid. In the next step, another enzyme phosphatidic acid phosphatase will work. Phosphate molecule is removed. So now glycerol only having the 2 acyl group on a first and second carbon, and this is called the 1,2-diacyl glycerol. On a last stage, 1,2-diacyl glycerol is converted to triglyceride by adding of the another fatty acyl CoA by the enzyme 1,2-diacyl glycerol acyl transferase, and triglyceride get formed. Now we just try to concentrate how that fatty acid synthesis going to affect when we are in a well-fed condition or when we are in a fasting condition. So let's see this figure, and we start to understand this figure with the glucose molecule. Now we all know when we are in a well-fed state, and most of our food having the 60 to 70% is carbohydrate, carbohydrate is there, that glucose will goes in a glycolysis, converted to pyruvate and ultimately converted to acetyl-CoA. Once this acetyl-CoA is more in amount, some portion might be definitely goes to the TCA cycle, but when there's a more in amount is there, that more fatty acetyl-CoA might go for the formation of fatty acid synthesis. Another reason behind is that when you're taking at a high glucose level, already insulin level is very high, and that high insulin level will induce the fatty acid synthesis. And we know that once the fatty acid synthesis is there, we having the high fatty acid level, it will help for the triglyceride synthesis. So we can say in a well-fed state level, when high insulin is in a high level, we are having the high triglyceride level, and this will happen into the liver. Once having the high triglyceride level, the triglyceride level has to be stored into the adipose tissue, and for that purpose it has to be transported to adipose tissue. So triglyceride is not transported directly to the adipose tissue. First of all, in a liver, it will convert it to another lipoprotein, that is a VLDL, very low density lipoprotein, and through the VLDL is reached to the adipose tissue, and then after it will remain into the adipose cell. So this is about the well-fed condition, where pathway will work like this. So we can say in a well-fed condition, when glucose is high, when insulin is high, at that time it increases the triglyceride level, increase the VLDL level, and if any excess calorie is there, more amount of the adipose tissue get stored. More amount of triglyceride gets stored into the adipose tissue. Now we think about in a fasting condition. In a fasting condition, glucose level may be a very low, so body will try to do the gluconeogenesis. So when the gluconeogenesis process will happen, body always try to take the energy from the glycogen, and glycogenolysis will happen and it will form the glucose, and almost be having the enough glycogen storage to maintain the glucose level up to the 16 to 18 hours, and still person will continue with the fasting. That triglyceride will, which is there with the adipose tissue, will goes in a breakdown, and this breakdown is called a mobilization. That same thing we learn into the beta oxidation of fatty acid, that when we oxidation of fatty acid will happen, first step is that mobilization of fat, and that mobilization of fat, it means triglyceride get broken down and the glycerol and fatty acid get detached, and that fatty acid get transported to the liver, and then after that fatty acid will be goes into the beta oxidation, which is not clearly clear here, but it will goes in a beta oxidation and it will convert to an acetyl-CoA, and that acetyl-CoA goes to the TCA cycle and ultimately produce the energy. Same way, when gluconeogenesis process is going on, the acetyl-CoA, and when we are not in a well-fed condition, the acetyl-CoA coming from the fatty acid, but that acetyl-CoA cannot go into the TCA cycle, and that is why some amount of acetyl-CoA goes into cholesterol synthesis, some acetyl-CoA may goes into the ketone body synthesis, and they may be utilized by the brain. Obviously the brain can utilize the glucose also, but when the glucose is not available, then alternative molecule ketone body will be utilized. So in another way, we can see in a fasting condition, triglyceride is broken down when epinephrine and non-epinephrines are very high, glucagon is very high. At the time, this triglyceride is broken down. We have to understand about the triglyceride synthesis in relation with the diabetes mellitus. Like when the person having the type 1 diabetes mellitus, we knows that there is a absolute insulin deficiency is there, and we know that insulin is a one molecule which protect the fat, or you may say that it protect the fat lysis and proteolysis. So when it's deficient, definitely that protection will not remain. So ultimately, whatever the triglyceride is there is the adipose tissue, it get mobilized and start to get broken down. So ultimately, when the mobilization is there, more triglyceride is broken down. Ultimately, there will be an increased free fatty acid level. So more fatty acid will reach more fatty acid will reach to the liver. But when it's reached to the liver, as the insulin is deficient enough, triglyceride synthesis will not happen, because for the triglyceride synthesis, insulin is always required. That's a stimulatory factor, and triglyceride synthesis mainly happen to the adipose tissue, where those receptors are insulin dependent. So if the insulin is not there, triglyceride synthesis will not happen. If triglyceride synthesis will not happen, ultimately VLDL formation also get affected. While we discussing about the type 2 diabetes mellitus, some sort of difference is that with the type 1. In type 2 diabetes mellitus, we know that the main problem is there with the insulin receptor resistance, and that is why glucose is high, and because of the high glucose level, with the feedback mechanism, insulin level also remain high. This high insulin level will not help to protect the fat, because receptors are resistant. So mobilization fat will happen, same like type 1, increase fatty acid level, and that free fatty acid will reach to the liver. And now it makes the difference here. As the insulin is already high, and there is a high free fatty acid level is there with the liver, there will be more triglyceride synthesis in the liver, and triglyceride level get increased. So patient of type 2 diabetes mellitus may be having the hypertriglyceridemia. So that already patient having the fat with the adipose tissue, which is mobilized to liver, and in the liver it will form the circulatory triglyceride, and circulatory hypertriglyceridemia will happen. This triglyceride will definitely will be utilized for the formation of the lipoprotein, so there will be a increased VLDL synthesis. So patient might be having the high triglyceride level, high VLDL level, with patient having the type 2 diabetes mellitus, and that will increase the transport of triglyceride to adipose tissue to the VLDL. Now it might happen, and that may be one, may put the one hypothesis that because of that, type 2 diabetes mellitus may having the chances of obesity in long term. Hope you have enjoyed this lecture. I try to enumerate things very simply. I will upload this lecture into my Telegram channel. You can search in our Telegram with the name of Biomed Piyush Tailor. This is my Telegram channel name. where you find this PPT as well as the same video lecture. Thank you very much.